We are live now.
Good afternoon, everyone. Good afternoon, everyone. I'm Dr. Maunika. I welcome you all for today's session on behalf of Shield Healthcare. And today's session is going to discuss about sperm DNA fragmentation. And I welcome our dear speaker, Dr. Gayatri Satpathy. She is MBBS, MD, DNB, FNB, fertility specialist in Oasis Fertility, Bhubaneswar, and ex-SR IMS BHU, and ex-SR SGPGI Lucknow. She's a fertility consultant.
No, I'm ex-fertility consultant.
Sir, ma'am, sorry?
I'm an ex-fertility consultant at Nova. Right now, I'm at Oasis Fertility. Yes.
Yeah, okay.
Yeah.
I welcome you, ma'am. I welcome you for today's session. I hand over the session to you.
Thank you so much, dear Dr. Monica. So I shall start sharing my screen now. I hope my slides are visible.
Yes, ma'am, visible.
Yes. So good afternoon, everyone. And I thank all of you who have joined me. And it's such an interesting topic and a topic which is very much required to be discussed on regarding sperm DNA fragmentation. So particularly the relevant clinical practice recommendations is what that I shall be focusing on. So briefly about me, I am consultant fertility specialist at Oasis at Bhubaneswar, and I shall be starting now. So we know that there are presently around 180 million couples worldwide who are suffering from infertility, and male factor alone contributes to nearly 20%, and in combination with female factors, it contributes to nearly 50% of the cases of infertility. However, with time, we have seen that the incidence of male infertility is rising, and this is in parallel with a decay in the semen quality as well. As we can see in this study, which has been conducted over a long period of time from 1989 to 2005, the sperm concentration has been declining. And for this reason, even WHO had to update the semen analysis values, and now we have lower values limit, which has been set. So what is the reason for the rise in this male factor infertility? So one is the paternal aging, that is as in females who are because of career orientation, they're delaying the parenthood. Even males are also delaying the parenthood, and parental aging or paternal aging also can lead to impaired cellular functions. They become vulnerable to diseases as their age becomes more. Then there is modification of different hormones. So late onset hypogonadism, andropause may sometimes set in. Genomic instability is found with older males. The second is the lifestyle modification, which has also contributed to this rise in male factor infertility. Incidence of obesity is rising. There is increased alcohol and tobacco consumption, high energy intake, nutritionally poor food intake, then use of recreational drugs, then more stress and anxiety seen among the male partners. Apart from this, the pathological causes, as we all know, there can be genetic susceptibility, there can be sexual disorders, there can be testicular injury or any diseases, various endocrine autoimmune disorders, sexually transmitted infections, which can affect the testes or the epididymis. And the environmental factors also are responsible, like radiation exposure or tight-fitting underwear, recurrent hot bath or sauna by the male partner, exposure to various chemicals due to job-related profile, occupational exposure to various toxins, chemicals. So these all are contributing to a rise in the male factor. So in spite of the fact that the male factor is contributing to nearly 50% of the causes of infertility, and in spite of the fact that the male factor is responsible for the adverse reproductive outcomes we are seeing, even following ART, we are still the male partner evaluation is limited to only the conventional semen analysis, which remains the standard initial investigation and sometimes the only investigation that is done for male infertility evaluation. A semen analysis test only looks at the quantitative parameters, and even we have not much progress with regard to the semen analysis also over time with regard to any different parameters we are checking. It is only the same parameters, only the values are changing. However, we are not improved on the semen analysis much. But we also know that sperm DNA fragmentation is an important contributor to male factor infertility. but still it is not routinely assessed before or during infertility treatment. And a routine semen analysis, even if it shows a normal range semen parameters, that does not preclude that there would not be presence of elevated DNA fragmentation. So now we are going to discuss regarding the impact of sperm DNA fragmentation on fertility. As we all know, the normal sperm chromatin structure is essential for safe and healthy transmission of parental genetic material. So sperm DNA integrity is crucial for the fertilization process, the embryo development, for implantation, for ongoing pregnancy, and for a live, healthy offspring. The DNA damage to the sperm can be at various levels. There can be mitochondrial DNA damage, there can be telomere attrition, there can be sperm DNA fragmentation, Y chromosome microdeletions, epigenetic abnormalities, etc. But we are today particularly focusing on the sperm DNA fragmentation. The sperm DNA fragmentation can be of two types, depending upon the whether one or both the strands of the sperm DNA are affected. So we have single DNA fragmentation and double DNA fragmentation. Now, the causes for the sperm DNA breaks are broadly can be classified into three important causes, but they are also interrelated. The first important cause is the defective chromatin condensation. So during spermiogenesis, the bigger size spermatid has to get compressed and become a small sperm. And in this small sperm head, the whole of the DNA has to get compacted and fit in. So for that, the histones need to get replaced by protamines, and this abnormal histone-protamine ratio and also any DNA repair mechanisms which get, if it is itself is defective during the process of spermatogenesis and spermiogenesis, can induce DNA fragmentation within the sperm. The second is abortive apoptotic process. Now, during the spermatogenesis and spermiogenesis, a percentage of sperms are formed defectively, but these need to get screened by the Sertoli cell. Because the defective germ cells, generally they display certain molecules outside of them, such as example is phosphatidylserine, and also there is some signaling which happens between the Sertoli cell and the defective germ cell. So thereby the Sertoli cells are able to remove these the defective germ cells. They act kind of scavengers within the testes. However, either if the amount of defective germ cells become more, or if there is a defective signaling between the Sertoli cell and the damaged germ cells, that some of the damaged germ cells which contain the defective DNA, they escape the apoptotic process. And this escape mechanism is what is called as abortive apoptotic process. And the third one is the oxidative stress. That is when there is an imbalance between the reactive oxygen species and the normal defense antioxidant system which should be there within the seminal plasma. There arises an oxidative stress. So if we understand oxidative stress a bit more, so normally there should be a balance. Because why balance? Because some endogenous ROS is produced within the semen. So the source of these endogenous ROS are leukocytes which are present in the seminal plasma, the damaged spermatozoa which is present in the seminal plasma, and to balance this endogenous ROS, some amount of antioxidant is present in the seminal plasma. For example, we have catalase, glutathione peroxidase, superoxide dismutase, and various vitamins like vitamin A, C, E, etc. But when there is an excessive ROS, which is beyond that which the antioxidant system can control, which could be due to various reasons. For example, as I mentioned, the disordered lifestyle due to smoking, alcohol, a poor dietary intake, lack of exercise leading to obesity. So the hormonal imbalance within the males can also contribute to this. Then exposure to various environmental factors like exposure to pesticides or heavy metals, drugs, and sexually transmitted infections, and testicular sources, for example, if the patient has varicocele or cryptorchidism. So if all this is increasing the amount of ROS within the seminal fluid, so thereby there is an oxidative stress. And this not only impacts the nuclear DNA, but also the mitochondrial DNA gets affected. There is lipid peroxidation and abnormal apoptosis. But why are the sperms so susceptible to oxidative stress? They are so much susceptible because of the abundance of the polyunsaturated fatty acid which is present in the seminal, in the sperm plasma membrane. And also the the amount of antioxidants is lesser within the cytosolic content of the sperm. It's most of the antioxidants are present in the seminal plasma, but within the sperm head, the quantity is less. And the sperm possess very less DNA damage detection and repair mechanism. So thereby they get more susceptible to this oxidative stress. So the oxidative stress thus damages the sperm DNA, the sperm protein, lipids, biomembrane, thereby impairing the sperm function and leading to male infertility. Thus, the variety of causes leading to sperm DNA fragmentation, either these are endogenous causes because of defective DNA repair mechanism or due to some endogenous problems arising within the males like the varicocele, or it could be external factors like the disordered lifestyle and smoking, etc. So ultimately, the sperm DNA fragmentation, if it builds up, it has got potential effects on the future reproductive outcome. It reduces the pregnancy rate in couples who are trying for pregnancy naturally also. It causes recurrent miscarriages. There is a suboptimal embryo quality, which formation and impaired fertilization. The ART outcomes are also suboptimal, whether it is IUI, IVF, ICSI. And it is also found that the children who are born out of men who have higher sperm DNA fragmentation in the semen may suffer from future genetic diseases and childhood malignancies as well. So the impact of sperm DNA fragmentation on the reproductive outcomes, it actually depends on a balance. The balance between the amount of DNA damage which is there within the sperm head and the DNA repair capacity of the oocyte. But when the oocyte's repair capacity is lesser than the amount of sperm DNA damage, then it influences the embryo development potential and the health of the offspring. So as a result, the resulting embryo may fail to develop, or it may fail to implant in the uterus, or it may get aborted naturally at a later stage. And the sperm DNA fragmentation may not be perceived at the time of fertilization, but because generally there is a late paternal effect. It is because of the late paternal gene expression, which usually happens when the embryo is 4 to 8 cell staged. So what is the rationale of sperm DNA fragmentation testing? It is to attain more in-depth knowledge about the sperm quality because of the critical function of the sperm DNA integrity for a successful reproductive outcome. So what are the current recommendations for sperm DNA fragmentation testing? So according to the societies like the American Urological Association and the American Society for Reproductive Medicine, the routine use of sperm DNA fragmentation testing is not recommended during a routine infertility evaluation. However, the ASRM 2015 Practice Committee has acknowledged that varicocele repair and antioxidant use might be of value to reduce the sperm DNA fragmentation. Testing for sperm DNA fragmentation might be clinically informative for IUI, IVF, and ICSI outcomes. And it also acknowledged that spermatozoa, which are retrieved directly from the testes of men who have elevated SDF in the neat semen, could have better DNA quality than the ejaculated counterpart. So what are the different sperm DNA fragmentation tests? So we can broadly classify them into direct fragmentation tests and indirect fragmentation tests. So direct tests are those which are directly measuring the extent of sperm DNA damage by using certain probes and dyes. So these probes are directly getting fit into the site of the DNA breaks, and we can have a quantitative measurement of the amount of DNA damage which is actually present in that sperm. But the indirect tests are those where they are measuring the susceptibility of DNA to denaturation. So denaturation can be done with acid or alkali, etc. Why? Because those DNA which have got fragmentation within them, so they are more susceptible to get denatured in comparison to normal DNA. So most of the tests are indirect tests actually, SCSA test, SCD COMET. So SCSA is sperm chromatin structure assay test. SCD is sperm chromatin dispersion test, and COMET test, which is also popularly known as the halo test. So what are the current recommendations for the SDF testing procedure? So this is the latest clinical practice recommendation published in Andrologia Willi publication journal 2021 and by group of Esteves et al. So according to this latest recommendation, the most reliable tests for assessing sperm DNA fragmentation are SCSA, alkaline COMET test, SCD, and tunnel test. However, a standardized protocol is required with strict quality control is essential for a reliable SDF testing result. So tests should be validated by the lab and with thresholds established based on evaluation of both the fertile and infertile population. So the recommendations are you can use either of the any of these tests, SCSA, tunnel, SCD, comet, but the test need to be done on a neat semen sample, and which has been obtained after 2 to 5 days of abstinence, and validated protocol needs to be established, and we need to have our own laboratory thresholds. But in general, if we speak about the thresholds, if it is the DFI levels are more than 15 to 25%, you can inform the couple that they have an increased risk for male infertility, and particularly if the values are more than 20 to 35%, the chances of pregnancy, the probability decreases, even with the ART techniques also. So any testing we are doing, we need to understand the limitations regarding what they are actually informing us. So the SDF testing, it cannot tell us for sure whether a man is fertile or infertile, or it cannot say, okay, these particular couple are going to have successful outcomes out of IUI, IVF, or ICSI, or these particular couples are not going to have a successful outcome. Because the usefulness of any test for any one partner is also dependent on the fertility of the other partner. So what are the current recommendations for indications for SDF testing? Again, I have quoted from the latest clinical practice recommendations by Steves et al. So the first important indication is in men who have varicocele. So men with varicocele who are seeking fertility, they should be informed that varicocele may cause SDF, and that repairing a clinical varicocele can elevate the SDF. Potentially increasing the likelihood of reproductive success. And then there are certain men with varicocele where clinically the varicocele is there. However, the semen analysis is normal or borderline. The men do not have any symptoms. So most of them, we do not understand whether we should go ahead with varicocelectomy or not. So in such situations, the SDF testing can be of help if men who have high SDF levels in such a scenario should be advised to go for a varicocelectomy, because it has been seen that post-surgery the SDF levels come down. But the repeat levels have to be done at least after a break of 3 months. And the current guidelines with regard to subclinical varicocele is that SDF testing is not recommended in this particular group. Now, the second group is the unexplained infertility patients who have idiopathic infertility and recurrent pregnancy loss. Again, you can inform these couples that SDF is also one of the contributory causes to this, because many times we do a battery of tests and we do not actually find any cause for unexplained infertility patients and recurrent pregnancy loss. So maybe for explanatory purposes also it can be done. And in case we have found somebody who has high SDF in such a scenario, we should go for a prompt complete male evaluation so that we can identify and maybe we can treat the condition, which can reduce the SDF, and probably then the couple can have a better reproductive outcome. And the third is men who have infertility risk factor. So there are many people who come to us for the fertility counseling. They come for pre-pregnancy screening. So and among such couple, those men who have risk factors, like they have history of tobacco exposure, smoking exposure, they have obesity, they have metabolic syndrome, or they have exposure to various because of their occupation, they are exposing to various toxins, drugs, and or advanced paternal age. Again, they should be informed that such men, they have risk for high SDF. If you screen for men with high SDF and with lifestyle modification or by giving certain medication, we can even increase their likelihood of future reproductive success. So to summarize, so these are the indications for sperm DNA fragmentation testing. One is men who have varicocele, who have unexplained or idiopathic male factor infertility, and couples who come to us with recurrent pregnancy losses. For IUI, IVF, ICSI, we can even offer them in the beginning itself, even for those patients who have not undergone IUI, or you can also offer them to patients who have a repeated IUI failure, repeated IVF failure. And suppose a patient you have selected for IUI but has got very high SDF based on the risk factor. So you suggested SDF testing and it came out to be very high. So maybe even you can counsel them that early ICSI is better rather than going for IUI, because chances with high SDF, the chances of the IUI pregnancy rates are lower. And then patients who are coming to us for fertility screening, and then you identify risk factors, so then you can advise them to go for SDF testing. And then sperm freezing. Men who come to us for sperm freeze generally come for fertility preservation. So these patients generally have some sort of cancer, or they're exposed to some chemotherapeutic agents. So they as such have a high risk for increased SDF. So testing for SDF and then going for sperm freezing would be helpful, because later on when you would require this semen for the fertility purposes, the SDF SDF levels also can guide you whether it is better to use this semen for IUI or is it better to directly go for an IVF ICSI. And also counsel the patients that sperm freezing itself can also increase the sperm DNA fragmentation to some extent. So any patients we are identifying with high SDF level, always better to go for a male evaluation, identify factor, treat the underlying factor. Along with that, give them good lifestyle advice like quitting smoking, reducing weight, avoiding the wearing tight underwears and hot water baths. And so some simple lifestyle advice can also help them. And along with that, different antioxidant medications. And the other thing is that these patients who have high SDF, so if in case the SDF does not come down even after treatment, it is better to consider ICSI, particularly ICSI, because when we are doing an ICSI in comparison to conventional IVF, we are able to select the morphologically normal sperm, and these sperms are supposedly having lower sperm DNA fragmentation within them. And also particularly those patients who have repeated ICSI failures, it is also advisable to go for testicular sperm extraction and using testicular sperm for ICSI rather than directly from the ejaculate sperm. So now we are going to discuss about managing the sperm DNA fragmentation. So as I discussed, it is because first is addressing the cause. So cause can be addressed only if we have a complete male evaluation is done, weight reduction. If varicocele is present, go for varicocele repair. Healthy lifestyle needs to be adopted, and antioxidants are an important part of the management of the SDF. So antioxidants can be given orally as either a single component or mixed component. The different antioxidants are obtained from dietary supplements like vitamin C, vitamin E, carotenoids, carnitins, ubiquinol, cysteine, folate, selenium, and zinc. Multi-antioxidant supplementation is considered to be more effective for male fertility parameter because of the synergetic effects of the antioxidants that we can utilize. Because different antioxidants work at different levels of the sperm, some at the head, some at the tail, some at the level of DNA. So we can utilize all of them, so the synergistic effect, and then we can have a better response. And any antioxidants we give, we need to at least wait for a minimum of 3 months, 2 to 3 months for the effect to be noticed. The Cochrane review, which was last published in 2014 about the effect of antioxidants, so they included around 48 randomized control trials, and which had around 4,179 subfertile men. So the Cochrane review was that there is a considerable variability in the antioxidant effect on semen parameter, but they reported that there is a statistically significant improvement in the live birth rate and clinical pregnancy rate. And there is a growing body of evidence, multiple meta-analysis, systemic reviews, they're all mentioning that there is a beneficial effect of antioxidant use in infertile men with sperm DNA fragmentation. However, due to high heterogeneity is because of multiple studies are using different antioxidants at different doses, some single, some multiple. So because of this, there is a high heterogeneity among the studies and low quality of methodological techniques. So because of that, we are not able to conclusively say that there is a definitive effect. However, despite the lack of strong evidence, use of antioxidant is still considered a valuable option for treatment of infertile men with sperm DNA fragmentation, either solely or as a part of the treatment strategy, which is individualized to each patient. So today's take-home messages are, although the routine use of SDF testing is still not widely recommended, in selected group of patients, SDF testing can provide complementary data that cannot be provided by traditional semen analysis. So it can give you independent information about sperm quality, and thus its integration into fertility clinic, into our practice, can help us in providing better counseling, better diagnosis, and better treatment planning, eventually leading to better reproductive outcome. So male partner of any couple, infertile couple who is found to have elevated SDF should undergo a proper evaluation. You should rule out the physical factors, especially varicocele and other occult male factor, because any reduction in SDF can help a couple even achieve natural conception. It can increase the likelihood of successful reproductive outcomes with our ART techniques. So I thank all of you who have joined today and for this webinar. And I would like to thank the Shield Healthcare for providing me this wonderful opportunity to talk on such an important and informative topic. Thank you once again. I am open to any questions if there.
Thank you so much, ma'am. Thank you so much for your time. And ma'am, we have one question.
Yeah.
One question is, if we are considering to do SDF test, which procedure would you prefer, tunnel or SCSCA?
Yeah, so this is what one of my slides was about, that any of the 4 tests can be chosen. Whichever test is for which you are having a good validated protocol in your lab and you have established the threshold, you can choose. So between SCSA test, COMET test, SCD, and tunnel, you can choose. And you have to set your own thresholds.
Ma'am, we do have another question too. Like, what kind of lifestyle modification should we counsel the patient, ma'am?
Yeah. So lifestyle modification includes one is abstaining from any kind of addictions if present, because smoking and alcohol use have proven that they increase the ROS within the semen. Second is we need to tell the male partner to avoid using a hot water bath or hot sauna, because that also increases the testicular temperature, and avoiding wear of tight undergarments. So they should preferably use loose cotton undergarments. So these are, and also if they're obese, they need to work on that, because even in obesity, the testosterone estradiol ratio is also deranged. So even with obesity, you can have a good hormonal environment and ROS can be reduced. And so these are the important ones. And along with that, we can supplement it with the use of antioxidants.
Thank you so much for explaining in detail, ma'am. The one who have asked the questions, they would be getting a clear picture of the answer. And I hope we do a lot of webinars on Shield Connect with us. I hope we do lots of things to discuss on our page in further too. And thank you for connecting with us, ma'am. Thank you so much for supporting us.
Thank you.
Thank you for time, ma'am. And there are only 2 questions, ma'am. We don't have more questions.
Yeah. Thank you.